I'm Markita Landry. I'm an assistant professor in the Department of Chemical and Biomolecular Engineering at UC Berkeley. And today I'm going to talk about tiny things to engineer solutions to life's big problems, which is the theme of my lab that has been supported by the Arnold and Beckman Foundation through the Beckman Young Investigator Program. So first, I wanted to talk a little bit about the influence of Dr. Arnold Beckman's contributions to science. And as a basic scientist myself, being a physicist by training, my early work largely focused on understanding biomolecular interactions that give rise to life. However, I'm also cognizant of the fact that to make these measurements and to take these data, we need instrumentation that can really push the boundaries of resolution in terms of what it is that we're trying to measure. And one of the themes that I most appreciate about the Arnold and Beckman Foundation is a theme that science informs the development of technology, and then technology in turn informs science. And in this really nice manner, there's a nice interplay between the development of technology that allows us to learn more about the world that we live in. So examples such as the spectrometer and the pH meter, which are really conceived by Dr. Arnold Beckman, have allowed us to push the frontiers of knowledge in a way that we would not have been able to without the instrumentation and the technology. Now, personally, the Arnold and Beckman Foundation has had a pretty strong influence on my scientific career even before I was an independent scientist. So as I mentioned before, I got my BS in chemistry, my BA in physics from the University of North Carolina at Chapel Hill, and went on to do a PhD in chemical physics at the University of Illinois. Here I was studying biomolecular interactions with optical traps, again, looking at how these fundamental forces drive life. And a part of my work was carried out at the University of Illinois Beckman Institute, where molecular dynamic simulations allowed me to confirm some of the experimental findings that my lab had been making. In being able to merge tools from various disciplines through the Beckman Institute, I quickly realized that to really advance science in a notable way, it's necessary to have multiple viewpoints and also multiple approaches. And so when I went on to do a postdoc at MIT, I chose to push my own boundaries a little bit outside of basic science and into chemical engineering. And today, the lab that I run at UC Berkeley is focused on understanding how nanomaterials can be chemically tuned to make very small analytical devices and biodelivery devices, very much in the theme of the Arnold and Beckman Foundation of taking basic knowledge and applying it to learn more about biological systems. And so, as I mentioned before, my early work during my PhD and my postdoc involved largely the development of tools and the use of those tools to learn about the forces that drive life. And many of the developments in these microscopic approaches allowed me to gain an appreciation for instrumentation and technology development from an early trainee stage. However, I never envisioned that in my own scientific independent career, I would move away from physics and move very much into applied biology. And so here I'm depicting some areas that my lab has been working on in the past few years as part of the Beckman Young Investigator Program. Here on the left, we can see the probes that my lab has developed to optically image neurotransmitters such as dopamine. So here we can chemically functionalize the surface of nanoparticles to light up in the presence of neurotransmitters such as dopamine, put them into brain tissue, and then stimulate that brain tissue to release dopamine so that we can measure the kinetics of dopamine release and reuptake, and then see what the influence is of drugs on this process. Similarly, we can also chemically modify the surface of nanomaterials for the purpose of gene and protein delivery in plants. Here, this process relies on adapting nanomaterials such that they can be carriers of molecules that are important for genetic engineering purposes. And so within this space, genetic engineering of plants, a part of my lab looks at how we can genetically modify plants to make them more sustainable and higher yielding for better agriculture in the future. Similarly, how we can perhaps upregulate natural product production, which plants are natural producers of many of our pharmaceuticals, and also in the bioenergy sector. Now, genetic modification of plants is an area that has yielded many successes, specifically in the generation of crops that are USDA approved for growth, and some even for human consumption, that again are going to be more robust to a changing global climate, and also much more robust to or against pathogen infestations. Similarly, the development of golden rice, which has had international relevance, where the gene for or the gene pathway for vitamin A production is engineered into a strain of rice, and this nutrient is therefore engineered into a staple food that can help fight nutrient deficiency. Now, part of the issue with the generation of genetically modified crops is in their costs. So today it costs on average $140 or so million and an average of 13 years of R&D to create one genetically modified crop. And that is because existing tools lack efficiency and also have to go through a significant regulatory pathway to be accepted. And so my lab has developed these nanotechnologies that allow us to deliver DNA into plant tissues and allow the expression of a transgenic protein, here shown as green fluorescent protein. And so in these confocal micrographs, we can see the expression of this transgenic protein that was delivered not by a pathogen or a virus, but rather that was delivered by small nanoparticles that were chemically functionalized to carry DNA vectors that code for green fluorescent protein. One of the interesting features of this platform that we discovered is that the expression is transient, meaning that if we measure the expression of messenger RNA specific to green fluorescent protein, we see that it peaks at day 3, but subsides by day 10. And these findings could have some significant regulatory implications in the technologies that we use for developing genetically modified crops. Specifically, if we were to use nanoparticles to deliver gene vectors that express a nuclease system called CRISPR, we could site-specifically modify the genome of the plant in a way that allows the plant to be genetically modified via an edit to the genome, but without traces of transgenic DNA incorporated into the genome. And so the advantages here being both on the precision and the regulatory nature of these technologies as applied to crops. A second area that my lab has taken an interest to is in the development of analytical tools to study brain chemistry. Now, the brain is a really multifaceted organ in the body that has many different layers of information. For example, the structure and connectivity of neurons in the brain can tell us about how neurons are organized into circuits and how these circuits perform higher order functions within organisms. These neurons are largely electrically active, and this is how cells communicate. Neurons communicate with each other through these logic-gated signals known as action potentials, such that when a neuron fires, it can release information to neighboring cells through the release of chemical neurotransmitters, giving the cells chemical activity as well. So while the information that's gated through electrical activity gates the activity state of the cell, of the neuron, the chemical activity is really what drives the communication between cells. And for a long time, the analytical chemistry approaches that were used to measure chemical activity were far too large and far too clunky to measure this chemical activity at spatial and temporal scales of relevance. So this is known as a missing dimension in neurobiology, where being able to see the chemical communication between neurons in the brain is really an exciting new frontier in neuroscience. And this new frontier could have implications in how we study the effects of pharmaceuticals, especially those that target neurotransmitters and neuromodulators in terms of their release or their reuptake to modulate higher order activity. And so my lab has worked on the development of nanotechnologies where nanoparticles can be chemically modified to light up in the presence of neurotransmitters such as dopamine. These nanosensors can then be embedded into the extracellular space of brain tissue slices and then put into a microscope for imaging. And so what you're seeing here are the nanosensors embedded into brain tissue, and the brain tissue is being stimulated optogenetically or electrically to release dopamine. And as you can see, dopamine is released upon stimulation and is also reuptaken by the tissue in a way that is modulated by the strength or the intensity of that stimulation. Now, one of the questions that we're interested in pursuing further is the effect of drugs on the kinetics of this process, where we can add drugs to the brain tissue that would either increase or decrease synaptic dopamine to see how these changes affect brain tissue as a whole. So for example, if we were to add quinpirole to the bath, we would see that the effects of the drug are to attenuate dopamine release in the extracellular space, as we would predict, and that we can also do this in a way that queries not only the ensemble average response, as we're doing here with these traces, but asking what each individual hotspot in the brain is also doing upon response to these drugs. And if we start looking at the data from a point source by point source basis, we can see that while most drugs have the predicted response, which is to decrease dopamine release relative to the pre-drug condition, there's a significant number of putative dopamine release hotspots that behave opposite to what we would expect for this drug. And so the nature of this biological heterogeneity is something that we can now probe using a tool that has both the spatial and temporal information accessibility that previously did not exist. And so with that, I would greatly like to acknowledge the Arnold and Beckman Foundation for support of this work, and specifically the Beckman Young Investigator Program for allowing my lab to venture out of physics and out of biomolecular interactions as a field of study and into the development of nanoscale technologies and tools that will allow us to learn more and manipulate better biological systems.